Today we'll talk about the law of independent assortment. What this law of independent assortment is. This is law of independent assortment. This law of independent assortment. It is Mendel's second law of inheritance. It is Mendel's second law of inheritance. Mendel's first law of inheritance is law of segregation, and Mendel's second law of inheritance is law of independent assortment. And this law of independent assortment, it deals with two genes or two pair of alleles. As compared to that of law of segregation that deals with two alleles of the same gene. This law of independent assortment, it deals with two pair of alleles of two genes. Now, what is law of independent assortment? Law of independent assortment says that the inheritance of one character is independent of the inheritance of other character at the time of gamete formation. That means one character, when the inheritance of one character, for example, we have seed shape and seed color. These are two characteristics. They are transmitted from parents to offsprings. These two characters, they will assort independently of each other. They are not tied up together, but they will assort independently. They segregate independently of each other and will get transmitted from parents to offsprings. So this is what is called as law of independent assortment. That is, the inheritance of one character is independent of the inheritance of other character at the time of gamete formation. So law of segregation, law of independent assortment states that inheritance of one character is independent of other character at the time of inheritance of one character is independent of the other character in an individual. Inheritance of one character is independent of the other character in an individual, or we can say that, or the two genes assort independently of each other at the time of gamete formation. As this law deals with independent assortment, deals with two genes, so we can say that the two genes assort independently of each other at the time of gamete formation. If we have one gene, it will go from parents to offsprings in one way, and the other gene, that will go in the other way. That means these two genes have no connection, no association. They are not tied together. So the two genes assort independently of each other at the time of gamete formation. Two genes. Another statement: two genes assort independently of each other. Independently of each other at the time of gamete formation. So it means that the two genes assort independently of each other at the time of gamete formation. What it means? It means that when two allelic pair are combined in an individual, the segregation of one pair of allele is independent of the other pair of allele. This is also another statement for the law of independent assortment. That means we have two different pair of alleles, one pair of allele, and this is another pair of allele, and when they are combined individual, after that gamete formation will take place, their segregation will take place, and that segregation will be independent of each other. So we can say that this statement can be put in another way, that if we have got two allelic pair in an individual, if two allelic pair are combined in an individual, the separation of one pair of allele is independent of the other pair of allele at the time of gamete formation. Or we can say that when it's another statement, when two pair of alleles are combined in individual, the separation of one pair of allele is independent of the other pair of allele. The separation of one allelic pair is independent of other allelic pair at the time of gamete formation. So these are the three different statements of the law of independent assortment. Now question arises that this one chromosome pair assorts independently of each other, that forms the basis of the law of independent assortment. That means, for example, we have individual having genotype capital R, small r, capital Y, small y. Obviously, these are homologous chromosomes. This is capital R, small r. This is capital Y, small y. These are homologous chromosomes. These are homologous chromosomes. These are homologous chromosomes. This one chromosome pair assorts independently of other chromosome pair, that forms the basis of the law of independent assortment. That means this one chromosome pair assorts independently of other chromosome pair, forms the basis of law of independent assortment. Basis of law of independent assortment. It forms the basis of law of independent assortment. So we have gametes. We'll make gametes here. We have gametes. That means one chromosome pair assorts independently of other chromosome pair at the time of gamete formation, and that forms the basis of the law of independent assortment. So we have gametes like this. This is capital R and capital Y. As we see gametes here, we have capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So these are gametes. Same is the case here. Now we have shown it by means of chromosomes. That means this is capital R, capital Y. So capital R, capital Y. Now one chromosome assorts independently of other chromosome pair forms the basis of law of independent assortment. So obviously this is capital R, this is capital R, small y, capital R, small y. Then we have this is small r, capital Y, this is small r, capital Y. Then we have small r, small y. That means this chromosome assorts independently of other chromosome pair. This chromosome pair assorts independently of other chromosome pair. That forms the basis of the law of independent assortment. This is small r, small y. One chromosome pair assorts independently of other chromosome pair at the time of gamete formation. So obviously this is small r, small y. 4 type of gametes, 4 gametes. So this chromosomal separation, it forms the basis of the law of independent assortment. Now we have this law of independent assortment is explained by means of a dihybrid cross. Law of independent assortment is explained by means of dihybrid cross. In dihybrid cross, we take two characters. For example, here we take two characters. One is seed shape, another is seed color. So we have two forms of seed shape. One is round seeds and wrinkled seeds in pea plant. And seed color, we have two seed colors in pea plant. One is yellow seed color, yellow seeds, and green seeds. Remember that round seeds are dominant over the wrinkled seeds and yellow seed color is dominant over the green seed color. So we'll make a cross. So we have parental generation, pea plant with round and yellow seeds. So we have RR and YY genotype. This is phenotype. Obviously this is genotype. Same is the case, it is crossed with pea plant with wrinkled and green seeds. This is phenotype of the individual. What is the genotype? Small r, small r, small y, small y. Then we know that gametes, law of segregation will operate here. Gametes, that is meiosis. So we have gametes, this gamete, so capital R, capital Y, and capital R, capital Y. Gametes, how many gametes? Two types of gametes are formed. Then we have got F1 generation. Capital R, small r, capital Y, small y. This is F1 generation. This is F1 generation, first filial generation. And these are pea plant having round and yellow seeds. But this is heterozygous or hybrid because it is formed by a cross between two homozygous parents. Then we go for the selfing. Selfing here will F1 crossed with F1. So we have this type. It is crossed with the phenotype, with the individual which has same phenotype, capital R, small r, capital Y, small y. So then we'll go for their gametes, gamete formation. Gametes, we have capital R, capital Y, small y. It is capital R, small r, capital Y, small y. What would be their gametes? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are their gametes. Same is the case. We have capital R. We have capital R, capital Y, capital R, capital Y. Sorry, capital R, small y, small r, capital Y, small r, small y. These are the gametes. Then we'll go for the fusion of the gametes to get F2 generation. So to get the F2 generation, fusion of the gametes, we'll make a checkerboard. Fusion of this is male gamete, female gamete. The gametes, male gamete and female gamete. We have capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Same is the case. We have here female gametes, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So we have capital R, one gamete. These are male gametes, these are female gametes. Then their fusion, we'll see their fusion. This gamete will fuse with this gamete, so the individual have genotype capital R, capital R, capital Y, capital Y. This is capital R. This gamete will fuse with this gamete capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y. This gamete will fuse with capital R, small r, capital Y, small y. Same is the case. We have capital R, capital R, capital Y, small y. This gamete will fuse with this gamete, or R, small y, small y. This gamete will fuse with this gamete, capital R, small r, capital Y, small y. This gamete will fuse with this gamete, capital R, small r, small y, small y. This gamete will fuse with this gamete. We have capital R, small r, capital Y, capital Y. This gamete will fuse with this gamete. We have capital R, small r, capital Y, small y. This gamete will fuse with this gamete. Small r, capital Y, capital Y. This gamete will fuse with this gamete. We have capital R. We have small r, and capital Y, capital Y, and small y. With this gamete will fuse with this gamete. We have capital R, capital Y, small y. Then this gamete will fuse with this gamete. We have capital R, small y, small y. This gamete will fuse to this gamete. Small r, capital Y, small y. This gamete will fuse to this gamete. Small r, small y, and small y. This is F2 generation. Obviously, this is the genotype. A genotype of this. This is the genotype of the individual. What is this? This is round and yellow seeded pea plant. Pea plant with round and yellow seeds. This is pea plant with round and yellow seeds. This is pea plant with round and yellow seeded pea plant. This pea plant is also round and yellow seeded seeds. This is also capital R, capital Y. This is pea plant with round and yellow seeds. This pea plant is also round and yellow seeds. This pea plant is also round and yellow seeds. This pea plant has also round and yellow seeds. Because capital R, capital Y, that is round and yellow seeds. Then we have this pea plant has round and small y, small y. This pea plant has round and not yellow. This would be round and green seeds. Then we have round and yellow seeds. This would be also round and green, small y, it represents green color. This is capital R, capital Y, that is round and yellow seeds. This is small r, capital. This is wrinkled and yellow. This is wrinkled and yellow. This is also wrinkled, but this is capital Y, wrinkled and capital Y. It is wrinkled and yellow. Capital Y for yellow. This is capital, or this is round and small y. This is for green seeds. Small r. This is wrinkled and capital Y, yellow seeds. Then we have last one, small r, small y. This is wrinkled and green seeded pea plant. So we have phenotypic ratio. Phenotypic ratio is how many are round and yellow. That is, phenotypic ratio is 9:3:3:1. That means we have round and yellow seeds, round and green seeded pea plant, then wrinkled and yellow, and wrinkled and green seeded pea plants. How many round and yellow? If we'll calculate, they will come 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. This is round and yellow, 9 out of 16. Total is 4. 6 out of 16, 9 are round and yellow. Then round and green, out of 16, how many round and green? Round and green, 1, 2, and 3. That means 3. How many wrinkled and yellow? 3. 1 wrinkled, 1, 2, and 3. Wrinkled yellow, 3. So out of 16, 3 are wrinkled yellow. Wrinkled and green, that is out of 16. How many wrinkled and green? This is last one, wrinkled and green. That is 1. So phenotypic ratio is 9:3:1. So 9:3:1. 9:3:1. What is the genotypic ratio? What is genotypic ratio in case of diabetic cross? So we have, this is genotype. So it was 1:2:1, 2:4:2, 1:2:1. So this is genotype ratio. This 4, it is for example, we have capital R, small r, capital Y, small y. This is 4. Capital or small, capital, so this is 1, this is 2nd, this is 3rd, this is 4th. So like others, for example, we have 1 is capital R, small y, 1. This is what is called as genotypic ratio. This is what is called as genotypic ratio. See, in this case, when we made a diabetic cross, in the F2 generation, this is F2 generation. This is F2 generation, and in the F2 generation, we got new combinations as well as parental combinations. This is parental combination. We got parental combination. This is parental combination. This is parental combination. These are 2 parental combinations. In the F2 cross, we get 2 parental combinations: capital R, capital Y, and small r, small y, small. The occurrence of parental combinations, that is this one, because we have taken the parental combinations round and yellow seeded pea plants. This is round and yellow seeded pea plants, pure. And then we took another parent, that is wrinkled and green seeded pea plant. This is wrinkled and green seeded pea plant. So in the F2 generation, we got parental combinations. This indicates that the 2 characteristics, that is seed shape and seed color, they have assorted independently of each other at the time of gamete formation. If we will not get the parental combination, we would not have received the parental combinations in the F2 generation. In the F2 generation, we received the parental combination. But if we would not have obtained the F2 this parental combination in the F2 generation, it means that the 2 characters are somehow tied together. So the important point here is to remember that the occurrence of parental combinations. Parental combination is round and yellow seeded pea plants, pure. Rickled and green seeded pea plants, pure or homozygous. This is round and yellow seeded pea plants. This is rickled and green seeded pea plants. The occurrence of parental combinations. The occurrence of parental combinations in the F2 generation indicates that the two characters, that is seed shape and seed color, they have assorted independently at the time of gamete formation and they are not tied up together. So it means that the law of independent assortment applies where the two characters, they will assort independently of each other at the time of gamete formation. Now there is exception to this law, that is law of independent assortment, that is linkage. Linkage is exception to law of independent assortment. Linkage is exception to law of independent assortment. That means if two genes are located, if two or more genes are located on the same chromosome, they will be inherited together. They will not get assorted. At that time, law of independent assortment fails, because the genes which are present on the same chromosome, they will be inherited together from parents to offsprings. And the genes which are located on the same chromosome, they are called as linked genes. So law of independent assortment does not apply on linked genes. So linkage is exception to law of independent assortment. Thanks.